Having looked at the specific example of chymotrypsin in our last lesson, let's try to draw some general principles about enzyme mechanisms from that example. When we considered the example of chymotrypsin, we saw that it involved nucleophilic attack in two stages. Let's take a few moments to review the two types of nucleophilic substitution reactions. These reactions occur when one functional group is replaced by another as a result of nucleophilic attack, as outlined in the figure below. In this example, the functional group X is replaced by group Z. An SN1 reaction is substitution nucleophilic unimolecular. That is to say, the bond between X and R is broken first before the Z group is added. It follows first-order kinetics because it involves one molecule at a time. An SN2 reaction is bimolecular. That is, Z attacks the Rx bond, which is then broken. This would follow second-order kinetics since there are two molecules involved. This is the type of mechanism observed for chymotrypsin. In addition to this type of covalent catalysis, other mechanisms are common. General acid-base catalysis depends on the transfer of protons between an acid donor and a base acceptor. Perhaps you recognize that this type of catalysis was also apparent in the chymotrypsin mechanism, the histidine acting first as a base catalyst to extract a proton, and then an acid catalyst to donate it. Metal-ion catalysis is also called Lewis acid-base catalysis. This refers to the definition of a Lewis acid and base. In this definition, the base is the electron pair donor and the acid is the electron pair acceptor. Examples of Lewis acids and enzymes include manganese, magnesium, and zinc ions. An example would be that of carboxypeptidase A, which has a requirement of zinc for catalytic activity. In the active site of carboxypeptidase A, as shown here, the zinc ion is complexed to the imidazole side chains of two histidine residues and to the carboxylate side chain of a glutamate. The purpose of the zinc 2 ions is to activate the carbonyl group to prepare for acyl substitution. Although we see that there are a limited number of individual enzyme mechanisms, yet these may be combined in different ways, as in the mechanism for chymotrypsin.